So thank you. Indeed, I'm a PhD student at Wageningen University in Research, working on sort of combining different types of data in linear mixed models. And so today I wanted to show some things that I've been working on the past few months. So in terms of the structure, I'll just start out with sort of a little bit of background recap of genomic predictions, let's say in a single trial, multiple environments, and then multiple environments and multiple traits. I think most people here are quite familiar with that, so I won't take too long there. And then I'll actually show how I've been integrating these environmental covariates from, say, sensors, weather stations, or even satellites in the linear mixed model. And finally, I'll show some results from a real-world data set from Europe. So, I mean, as we all know, genomic prediction is essentially just predicting the, let's say, the genetic value of new genotypes as a function of marker scores. So if we've got the simple mixed model, we've got the fixed effects in X beta, random effects in ZU. And for this talk, it's mainly interesting to think about the distribution and more specifically the covariance matrix of the random effect U. So if we're talking about GBLUP, let's say within a single trial, obviously we've got a kinship or a genomic relationship matrix, and we multiply that with a single genetic variance. So our covariance structure is fairly simple. It's just one single matrix, so to speak. Now, if we want to extend that to multiple trials, obviously G by E becomes important. Well, we've seen similar figures a bunch of times already. There's multiple forms of G by E, of course. If you look at the left, we've got crossover interactions, so rank changes, but the genetic variances are more or less the same within both environments. On the right, we don't have actually any rank changes, but we do have this heterogeneity of variance. So, we do still have, let's say, genotype-specific adaptability to changing environmental conditions that result in these differences in performance between environments. And of course, we want to model that, and well, we can do that with a linear mixed model as well, in a pretty simple way, simply by replacing that single genetic variance with a genetic covariance matrix of the environments. Let's call that sigma E. And so we can model this in a bunch of different ways, of course. We can fit an unstructured model, a vector analytic model, or do some other things. But I actually wanted to structure this sigma E using known environmental covariables, temperature, precipitation, solar radiation, etc. And then once we do that, we of course just take the Kronecker product of the two, the kinship, and the sigma E to come up with the full covariance matrix of the random effect. Now, we can take that further and look at multiple traits within these multiple environments as well, or multiple managements within the environments. So suppose that we have different location year combinations, and within each of these environments, we're looking at an irrigated trial and a non-irrigated trial. That can be modeled as well. I think one thing that people often do is they sort of combine the management factor and the environment factor into a single new factor, and then fit, for example, vector analytic structure for that big covariance matrix. Alternatively, you can also, of course, take a second Kronecker product and simply model it using let's say, an unstructured correlation matrix or covariance matrix for the management, something for the environment, and then finally the kinship again. And then you end up with this double Kronecker product over there. And this is the actual model that I've been using, so this double Kronecker product. So how do we now take this idea of having these sort of separable covariance models and how do we integrate these environmental covariables into there? Well, we could do that with regression. I've decided to take these covariables and make a sort of kinship of the environments out of them. Well, why would you want to do that? I think there's a couple of scenarios where that would be interesting. The one that I looked at so far is, let's say, well, sparse testing. So if we're in a situation where we have on the x-axis environments, on the y-axis we have our genotypes. Let's say that we're in a situation where we have a small number of checked genotypes that are evaluated in each environment. And then next to these checks, we have unique sets of genotypes that are evaluated in each environment. Or perhaps, we could have genotypes that are replicated in a subset of the environments. I think that's an interesting sort of scenario where these environmental covariables could be useful, because this limited overlap, in my experience, does make it harder for example, a vector analytic model to accurately estimate the loadings and thus the correlations between environments. Another one, like Jip mentioned, of course, is prediction into new environments. I haven't looked at that so far, but that would be an obvious other use case of using these covariables. So how do we do it? Well, like I mentioned, I looked at a bunch of different ways, four different ways to be precise. Today, given the time, I just want to look at one of them, and that's using this non-linear kernel, you could call it, in a linear mixed model with multiple variances. And what I mean with multiple variances is that I want to, because I'm looking at management and environments, and I want to have a unique genetic variance for every environment within a level of management. Because I think in the past, when people have done this, with these kind of kernel approaches, often, the kind of limitation was that you would have to assume that there was a single genetic variance for all environments within a level of management because of this double Kronecker product. And we can do all of that using standard software. So more specifically, we only have to define an R function that computes the covariance matrix of, let's say, the managements and the environments given a vector kappa that contains our parameters, and we can choose those parameters ourselves. And this function also has to compute and return the partial derivatives of that covariance matrix with respect to all of the parameters that we end up using. And when we return these things and then simply pass them to ASRML and just leave all of the estimation to ASRML, so we don't have to worry about that, which is nice. So this nonlinear kernel uses, let's start with this linear kernel very shortly just to know what the difference is between them. A nonlinear kernel would simply use the environmental covariables to construct a correlation matrix of your environments, and that's it. So it's kind of similar to this kinship for the genotypes using markers. The difference with a nonlinear kernel is that we initially create a distance matrix of our environments, and we do that in this way. So suppose that we have environment I and j. We simply take the sum of the squared differences for all of the environmental covariables and then divide by W. So suppose we had temperature and precipitation. We take the squared difference in temperature, add the squared difference in precipitation, and then simply divide by 2. Of course, we would have more environmental covariables. Then in the end, we end up with this distance matrix, which is symmetric, obviously, and on the diagonal, it just has zeros, because the distance between an environment and itself is obviously 0. So that gives us our distance matrix, and then comes the nonlinear part, where we actually convert that distance matrix into a correlation matrix. We do that using this relatively simple formula, so we take e to the power minus h times our distance matrix, and that gives us our sigma e that we were looking for. Now, one result of doing this is that we can only end up with positive correlations. In my experience, I've mostly been looking at European data, so to be honest, negative G by E correlations, I haven't seen them yet, but that's one limitation to keep in mind. And on the right here, we have just a simple figure showing how these bandwidth values sort of transform these distances into correlations. So on the y-axis, we have the correlations that we end up with in sigma E. On the x-axis, we have the bandwidth, which is very similar to a variance, actually. It must be positive, but that's the only limitation. And the different colors represent the different distances. So, of course, if the distance between any pair of environments is very small, we can have essentially any bandwidth value, and our correlation is still going to be close to 1. Similarly, if the distance is large, our correlation is going to go to 0 very, very quickly as the value of the bandwidth increases. And for intermediate distances, you end up somewhere in the middle, of course. So once we have this, we can start putting it into our model. So again, I haven't written it fully down here, but if we just look at the distribution of the random effect, centered on 0, of course, we have this double Kronecker product with something for the management, something for the environments, and then the genotypes. And this becomes the following expression. So because I want to model multiple variances, that is a single variance for every unique management by environment combination, we need to estimate the correlations between management on the correlation scale. This has to be correlation matrix, CM. We take the Kronecker product with this nonlinear kernel that I talked about on the previous slide. Once we have expanded sort of that correlation matrix, we then pre-multiply with this SS transpose, where S is essentially just a vector of standard deviations that we also estimate. Once we've done that, we have our covariance matrix of the managements and the environments, and we then multiply with the kinship to again get our full covariance matrix. I already mentioned that we can do that with this function specified in R, given a vector kappa of the parameter. So in this case, suppose that we had two managements and two environments. As a simple example, we would end up with 4 variances, the correlations between management 1 and 2, and then of course this bandwidth parameter that we now actually estimate directly using REML. I mean, the idea of these nonlinear kernels is not necessarily new, but I think previously what people did was they used either cross-validation or some other approaches to, well, pick a value for this bandwidth. But I figured, well, wouldn't it be nice, because that takes quite a lot of time, of course, wouldn't it be nice if we could simply estimate this in the process of fitting our mixed model? And, well, turns out we can, of course. Graphically, it looks sort of like this, right? So we have the product, the variances, and then the genotypes again. So if you then look at a few results, I've just decided to include one data set. So this is the DROPS data set, drought-tolerant yielding plants. It's a data set with 2 managements, so irrigated trials and rain-fed trials, 14 environments in Europe, and I believe one of the 14 is in Chile, and 246 maize genotypes. And we have environmental covariables from weather stations, and I believe we have things like temperature, precipitation, vapor pressure deficit, etc. And because I was mainly interested in this sort of sparse testing scenario, I've said, well, let's say that we have a number of checks to sort of control the connectivity between your trials. And the non-check genotypes are going to be tested in 2 out of 14 environments. So if you then look here, I do have to apologize because I think I made this on a Monday morning and I switched around some of these labels. So I'll just ignore the legend, I'll just go through it myself. Here we have the results. So on the y-axis we have the Pearson correlation between, let's say, the blobs from this mixed model and the centered phenotypes of the test set. So we compute the accuracies within each environment, of course, and then take the average. So if we look at the black line, that's the main effect model. So it only has a random main effect for the genotypes, ignores G by E, purely looks at the additive model, so to speak. Then the blue ones are the vector analytic models, so those rely on the phenotypic data to estimate the correlations, and the darkest one is an FA1 model, the lightest one here at the bottom is an FA3 model. And then when we look at the other lines, we have the green and the orange one here, which are the single and multiple variance versions of this linear kernel, so just using a correlation matrix constructed using the environmental covariables. And here on the top we have green and red, which are kind of overlapping. Those are the single and multiple variance versions of the nonlinear kernel. So what's interesting is that, well, first of all, there's not that much G by E in this data set, unfortunately, but I think what's quite interesting is that this linear kernel approach, which is essentially, of course, a rich regression on your environmental covariables and markers, the products of those, it doesn't perform well. It outperforms the additive model, but it very quickly does worse than the standard vector analytic models, regardless of the connectivity between trials or environments. But if you look at this nonlinear kernel, which is, of course, because of this bandwidth, a little bit more flexible, we actually see that if there's, let's say, limited overlap between your environments, which would be hard for the vector analytic model to estimate, we do outperform the FA1 model slightly, up to about 40 checks, I think. That's where you sort of get this crossing over point, whether I guess enough information for the FA models to accurately estimate the correlations, and with these nonlinear kernel models, you're still kind of stuck to the structure that is imposed by this distance matrix. So we see something similar for the irrigated trials, except that there's even less G by E. I also looked at RMSE, and there we actually see more or less the same pattern, with the only caveat that now the multiple variance models actually do slightly better. So it does seem that if you have, let's say, large heterogeneity of variance, there is some extra value in actually modeling those unique genetic variances for all of the environments, instead of just assuming that they all have the same variance within a particular level of management. And on the right, the results are more or less similar as well. If you're interested, there's a bit more information here, also working on the paper, but that will take a little bit longer. And with that, I'd like to thank you, and if there's any questions, I'd be happy to try and answer them.